So, good evening, everyone. So, we're starting with the week 5 live session for the course Introduction to Proteomics. What we would be looking in this today's session would be mostly of the mass spectrometry based proteomics or MS based proteomics. Also, you can let me know that if you have any particular doubt or not. Otherwise, we can start today's session. So, here with that previously till uh, till particular previous class and session we had looked at different ways of gel based proteomics. So that is can talk about is that the to study the differential expression or abundance of proteins, right? So what we had done is that to understand the differential expression of proteins in particular through the method of gel we were separating it. So the idea remains the same that we need to separate the proteins. So why separation is required? Why is separation? So separation is required to identify each particular protein. So here whole concept of this study is to find out each particular protein. So particular spot in the gel was corresponding to a particular protein. So what is happening is that to each particular protein, for each single protein, how much is the difference in its abundance? How much is the difference in abundance, right? So how much is the difference in abundance or its expression level is the one which is required for us to understand at the level of the differential expression of proteins or abundance based proteomics, what we can talk about. Now, the same principle here is that we were separating the proteins, complete proteins in the gel through its properties of the protein. One is through either through molecular weight or through PI, that is its isoelectric point, and molecular weight. So those both of those things is what is generally was separating it so that we can get easily one particular protein spot. Otherwise, when we used 2D uh, dyes, that is for the differential in expression. So there, what we were doing is that we were uh, tagging the proteins with different dyes for different conditions, and finally measuring the intensity across. Now, this process, you know that it it is the one which goes through certain limitations in terms of scaling and in terms of uh, robustness and the reproducibility with the challenges laid into the data analysis. So the scalability of the gel-based proteomics is a challenge. Then we need to have the data analysis because the gel to gel variation is one which is very much prominent. So these need to be reduced because and then the data analysis and understanding the particular spot how much to allow. So we have already discussed like the saliency and other features that how much we should allow for certain proteins to be which spots to be taken considered as a uh, particular spot of a protein and which does not need to be identified as a particular spot. So all these challenges are what coming up in place to detect the whole proteome and to understand the different level of expression of the proteins as a whole. So here came the sophisticated instruments like mass spectrometry, which help us and which is still helping us to enable the rapid identification of the whole proteome to have a wider proteome coverage with much lesser sample preparation and lesser variation technique, and finally measuring the intensity of the proteins. So today we will be mostly looking into it in continuation with your this particular week task, that how what are the principles of mass spectrometry and how these ions and the proteins are we finally getting to identify. Okay, so fully and clear. Okay, so now what we will be doing is that, yeah, so just yeah, yeah. So now what is our particular principle? So here, if you see that it is simplified version of particular mass spectrometry, if you see that it is just we need to have a simplified where the sample gets injected, and after injection then the procedure remains the same, that is it will be separated, and after traversing a particular distance or particular separation particular time it will be get uh, analyzed in the detector. So the basic principle or idea, that is the separation of the proteins to identify each particular single protein, remains the same, but the principle and the procedure remains quite different, and it had led to a huge increase in the coverage of the identification of the proteins for a particular proteins, and it is helping us a lot in getting the uh, particular identification and quantification of certain proteins, right? So this is the one which is helping us to a great extent. But today, what we will do is that we will look into uh, much detail that about how that particular thing happens and what can be the acquisition parameters and what can be those particular things that to be taken forward for consideration. Okay. So from the gel-based simple techniques of characterization of the protein, so now we are taking the proteins at the ions level and separating and detecting it, and finally uh, correlating with the intensity of the proteins or the abundance of the proteins that is present in the different conditions, and try to understand that what is the differential level of the expression of this particular protein. So what is the particular role? So here, if we now talk about, so the you know that this particular tissue or any particular biospecimen for that matter will contain a complex mixture of proteins, right? So there will be a huge amount of protein. So from here, if we want to identify particular protein, it will be a lot more complicated, and it will be a lot more complications will be there. So first thing that is done is that in a general procedure, which we will again see that there are two kinds of things. So the basically the one which is widely used and is the most robust one we can talk about is the bottom up proteomics, right? So bottom up proteomics. So what we are doing is that from the tissue we are extracting the proteins. So first of all, proteins are being extracted out. So you need to extract the proteins. So now there are different methods for extraction of the proteins, and with the different biospecimens like the tissue and the if you have the animal cells, you have the tissues, you have the serum, plasma, or if you biofluids, or if you are working with certain plant, bacterial or fungi cells, fungi things, all will need a separate kind of treatment and separate kind of technique, which you will be using like mechanical uh, things, chemical degradation of the cell walls, and you will be furtherly extracting out those particular proteins. So after you do that, extract out those particular proteins, what you are going to do is that digest those proteins using different enzymes, uh, proteolytic enzymes. So we were getting the peptides. Then further, the peptides will again be comprised of a complex mixture of peptides. Again, if you are just injecting all these peptides at a go, so what will happen is that the detector that is present that will get overloaded, and it will not allow you for specific identification of the protein. Many signals will be lost. So the single caveat for this kind of method would be that many signals will be lost. So if your signals are lost, so then what will happen ultimately is that we will not be able to get those particular uh, data. So now that what happens is that first we need to fraction those proteins out. So through different liquid chromatography based technique where the peptides get phased out or the separated out. So peptide gets phased out or separated out so that we can reduce the complexity. So this process generally what it does is that reduces our complexity of the mixture. Then finally we can ionize it. So we will be looking in some moment about the ionization procedure and how it is done or why it is done. Okay, so how it is or why it is done. So then what we are going to do is that we can use it in the mass spectrometry to get the final result. And this spectra, so again here you will see the term like MSMS spectra. So what does it mean? We will also discuss it in some time as we move along. So we will be seeing taking that as well, right? So this is actually one kind of ions which generally looks like. So if you don't understand, it's fine. So this is just you can understand that it is a peptide bonds. If the peptide bonds are get broken in the C terminal, it is a Y ion type, and in the N terminal, it is known as your B ion type. But you can go through this particular book, Analysis of Proteins and Proteomes by Mass Spectrometry, as well as you can go to, there is one book known as APC and XYZ of peptide sequencing. So that book also is very, very useful. You can go through to understand the basics of how this particular is working. But yes, we will be discussing it in a little bit detail as we move along in today's session, where we'll be discussing. So I think this particular thing is clear, right? So we are first extracting out the protein, then the protein is a complex mixture, then we are digesting it through proteolytic enzymes in the method known as bottom-up proteomics. Of course, we will be looking into it in coming in some slides about the detail of it. Then what we are doing is that we are fractionating it so that the peptides are separated out and phased out. And after this phasing is done or separated, then the ions are formed, and we will also look at the ionization procedure and how what is the difference between the hard ionization and soft ionization techniques, and how this field emerged as a result of the invention of the soft ionization techniques. Otherwise, it was all the gel based and other rudimentary methods for the which was based on the physical properties of the proteins or molecular properties of the protein, followed by getting the spectra, and why this kind of tandem mass spectra we would be looking into it in detail as well. So now we will be moving on. Yeah, so please let me know if you have any doubt or you face any difficulty in understanding any particular thing. Okay, so now, so I have just briefly talked about this terminals, these ions, the types of ions and all. But yes, definitely, once we move ahead, then we can talk about in detail about those ions, and then how those are identified and how we can get deduce the sequence of the peptide that we will be talking in some time. So if we talk about basic skeleton of this mass spectrometry technique or mass spectrometry, so first what we told is that the creation of the ions, separation of ions and detection of ions. So if we take the analogy from the gel, it was all about the protein separation. So if we talk about the 2D separation and take the analogy from there, so it was that the protein will be separated on the basis of their pH, on the basis of the isoelectric point. Then it will be separated further on the basis of our particular, what I can say is that particular separation will be on the basis of the molecular weight. So here, first of all, the proteins or the peptides mixtures are created the ions. So before this process, so before this process would be the protein extraction, would be particularly protein. What I can talk about is that protein extraction, then digestion of peptides, and finally moving it. So from here, what we are doing is that the creating the ions, and then that ions needs to be separated so that the detector can detect. So this detector can detect maximal signals. So it can be able to detect the maximum signals possible. So all these can be detected in our particular to our particular technique. So that's why we need to separate it and then finally detect it. So now, what is the ionization procedure? So that I mentioned just now that we will be looking into it detail, the different ionization techniques and what is the ionization procedure and how this completely revolutionized the field. Otherwise, the concept of mass spectrometry or particular these things wouldn't have evolved so much if we wouldn't be allowed, if we couldn't create these particular ions and those separation, because this is so if I so we will also look into it, the what is the basic fundamental uh, theory or concept behind mass spectrometry, that we just it nonetheless just only detects the m by z values, which would not have been possible without our particular which this would not have been possible, actually possible, if we could not, if we hadn't used this particular technique. Because if the ionization was too hard and if we would have lost our actual sample, then what would happen is that we could not use this technique for identification and quantification of proteins. Now, what is the ionization procedure? So these electrically charged molecules, so when we have the electrically charged molecules, ions are accelerated by an electric field. So their speed depends on the ion's charge and weight. So just understand. So when ions that are electrically charged molecules, these are accelerated by an electric field. So their speed depends on the ion's charge and particular weight. So by measuring the time it takes for the ions to pass a certain distance, the incidence of different molecules in a test can be determined, so that how much time it takes for it to reach. So it was impossible, however, to use this technique on large molecules such as proteins, as I was mentioning before. Large ions could be produced in gaseous form. So this was not going to be used if ions could not be produced in gaseous forms. So this was done by Koichiro Tanaka, that laser pulses could blast apart so that the ions in gaseous form are produced. So it was Koichi Tanaka that showed that the laser pulses could blast apart protein molecules in gaseous form and are produced. So now there were actually much before our whatever technique or for mass spectrometry of the biomolecules like protein, it was used only for chemical molecules to identification for the identification of unknown chemicals. So it was used, like instance, for identification of unknown chemicals. So that is the identification of not the known, rather the unknown chemicals we can talk about. So the identification of unknown chemicals, their molecular weight was being used heavily. But these hard ionization techniques were those that resulted in large number of fragment ion and number of molecular ion is low. So this was not able to work for our biomolecules, and then came that we can have the soft ionization techniques. Thanks to them, you can see in the left hand corner, John B. Penn, Koichi Tanaka, and Koto Trich, who can, which this low energy resulted in low fragment ions and high molecule ion. And of examples are like ESI and multi, that is electrostatic ionization. and MALDI, which we use to a great extent. So now if we move on, yeah. So yeah, hello, if you have, do you have any question? One second, one second. Hello. Yeah, Akash, you can go ahead if you have any question. What is the meaning of the difference between the? Okay, so here it is known as that if you make your molecule that will be there, so it will be uh, first of all it will get into the fragments and then it will be ionized and then it will be uh, separated. Now, if you have a so what it's telling is that the particular ion, if it is a hard ionization technique, then the number of fragments will become very high, and each those ion molecule total molecular uh, that is your time, I mean the charge and the weight would be very low. But if it is a low energy one, then it will give to low fragment ions. Means what it is the meaning that if you have a one ion, one molecule that will be fragmented to, let's say, two ions, two fragments, sorry, two fragments. So their charge and that is your particular, what I can say, that your weight will be not so much low. Understood what I can explain you? Yeah, yeah, yeah. So this is the thing. So if your fragment is very, very low, then particular molecular ion will be very, very low weight, right? So if it is break into two, then it will be have some particular edge. Yeah, yes. This light? Yeah, this one, reverse phase. Yeah, so this is reverse phase liquid chromatography, of which we will come in detail also. Yeah. So any other question? I'm extremely sorry. I think there was some technical issue. I was not able to uh, uh, hear properly. Yeah. So please let me know if you have any other doubt till now. Okay. Okay. So this is the way procedure it moved ahead by having the ionization. That is that we could control the energy. So they could control the energy. Otherwise, the ionization techniques was very high and robust for biomolecules to be used. Okay. So now basic, what is the mass spectrometry principle? So simply it is the thing that it measures the M by Z. So basically it measures nothing but measures the M by Z, that is for our proteins identification. So just try to visualize it. For proteins identification, what it is happening is that we have, for instance, we have a peptide. So you have a sequence of amino acid, for instance, A, T, B, L. So this is a peptide sequence, right? So this peptide sequence will have its particular molecular weight. So let it be, for instance, 210 kilo daltons, anything we can think of. So these, whatever be the molecular weight, you know that with the particular uh, fragment of the, of course, by taking into account the isotopic and those things. So this, so what will happen if it is a particular peptide sequence that is being injected in the mass spectrometer? Then what it will happen is that it will be detected, and what mass spectrometer will give us the mass per charge ratio. Okay. So now if we know the charge, so for instance, the charge can be 1 or 2. So we are getting this particular peptide's molecular weight. Okay, so this is all what we can deduce. So nothing more we can deduce by mass spectrometry. Of course, that's why I told that we are using like MSMS, you can see here. So this is what required. So that is the particular MSMS picture. So why this MSMS we would be going uh, to some extent in today and of course in the next sessions. So this is why, because this just gives us a particular molecular weight of our protein. So what we are getting is that we are getting just the particular molecular weight of the proteins. Now, so here, if you do this simple sum, you can understand that whatever be the charge, so mass, so when m plus h ions is there, so it will be mass plus 1 divided by 1, m plus 2h will be mass plus 2 divided by 2. So because of the mass of the ion, mass of the H plus will also get added and then divided by 2, that is the charge 2, because it is a charge 2. So M plus 3 H plus plus or mass per charge. So this is separated according to the ion mass and the charge of the ions. So among the ionization procedure for MALDI, that is the matrix assisted laser disruption ionization, this produces only singly charged ion species, which results in your large M by Z ion species. Okay. So it just produces, always produces a singly charged ion species, whereas ESI can produce doubly or triplet charged ion species as well. Okay. So this is the basic simple procedure of the mass spectrometry. So this is not only for your proteins or particular uh, anything to do with proteins as such. So it is the basic principle of mass spectrometry that takes that just is able to calculate the M by Z ratios of any metabolite or any compound or any peptide or any protein. Okay, so this is how this thing is generally done. So this is the basic principle of mass spectrometry. Yeah, so now if we talk about MALDI, right? So what is this MALDI? So we have told that the MALDI is particular, uh, what is the full form? Full form is that matrix assisted laser desorption ionization, right? So it is matrix assisted laser ionization, desorption ionization. So what happens here is that you have those sample matrix. So in that sample, what you do is that you have the samples, then so just yeah. So then you have the sample, then you create one matrix, okay, with those analyze that will irradiate the ions. Then you have the cation, anion, and then the vacuum. So what you do is that you have the sample matrix before your laser irradiation. The sample and the laser will be hitting the beams, and your particular molecule will make the charged species, ion species, and it will uh, go to the your mass spectrometry for separation. So this is a process of separating your ions. So here, as we have mentioned, that it will only produce the singly charged ion species. Yes, it will just produce your singly charged ion species. Okay, so this is how it is generally worked. But of course, we can have a look at this particular link to know if we are interested to know more. If you are interested to know more about it, so let us just quickly. Yeah, so just let us quickly check with this. Okay, so the top image we will be coming in some to some extent to very quickly. We will be looking to it. So the top is something different that it is required, but here if we can see. Okay, so this from this video it becomes quite clear that it is not only the proteins, but any particular molecule. If you see that any particular compound, if you want to know that what it contains, which can be analyzed through mass spectrometry. So it is a very powerful technique and is also being used heavily in different pharmaceutical industry and different chemical industries as well. Okay. So what happens is that so you have the different compounds. Then what you are doing is that so you can see in the year 1970. So it was a GCMS, which is a different thing. Then came the multi-tube. So the principle for multi remains the same. Only the samples and the analyze you need to choose it. Yes, you can see. So structural analysis of biological macromolecules. So for the development of methods for identification and structure analysis. So Koichi Tanaka was awarded the Nobel Prize in the year 2002 when the ionization energy could be controlled and there was no such requirement for the hard ionization technique. So it was simple idea that the sample can be added in a matrix and then the laser can be radiated for separation of the ions or start of the ions. So this is a good graphics to see. So founded this Shimatsu. So sorry, so this the matrix multi MS, and this is shown by the Shimatsu, thanks to them. So this is how ultra early detection of the diseases and analyzing molecular structures can be done through MS. So we will be looking into the MS based analyzers in certain method, but here you can see that there are different uh, novel mass spectrometry methods came up. So MSN means the MS after MS, how many fragmentation is done. So we will be seeing that, of course, today and as well as in the upcoming lecture. So it will become much more clearer that why it is done and how it is required. So see, this is a liquid matrix. So you can see these papers also you can go here. So like 3-aminoquinone, P-fumaric acid as a multi matrix for glycopeptides, carbohydrates, and phospholipids. Ionic liquid matrices optimized for multi-MS, sulfated, saturated neutral oligosaccharides, and glycopeptides. Okay, so this is finally getting released. So this is the amyloid MS. So A beta related peptides by your antibody beads. So this is the example of magnetic using magnetic beads. So this is the case study and example that how this could help you to understand the amyloid beta thing for the Alzheimer's disease and for the amyloids. So you can see these are few examples which are showing the applications. Okay, so uh, any change in the mass for example here there was a change in the shift of the mass shift of 28 Dalton could be we can identify through the MS based instrument because it anyway it is the one which just takes into account mostly the our particular uh, uh, our particular mass of that mass charge ratio. Okay, so these are some other techniques. So you can, of course, go ahead and look into it. So I can paste it in the chat box as well for further. If you have any further query, you can refer to it. So I'm just pasting in the chat box as well. Okay, so now we will be moving into it. Sorry. Yeah, yeah. So now the second method which we have talked about is the electrospray ionization. So that one thing which we talked about is that the Maldi and it was like Koichi Tanaka and all those things helped us in understanding. But the problem is that what we talked about is that it can produce only singly charged ion species, right? So if it is producing only singly charged ion species, then what happens is that it will not be able, will have the M plus H pretty high. So if so, for instance, we will be talking with the different mass analyzers, but as in that particular article and the video which we were seeing, so it was telling that the principle of multi-top, right? So if I talk about multi-top, so top is nothing. That is, it will calculate the time we will have in few slides about the principles in great detail. Calculate the time of flight of the particular ion, right? So particular ion when it will be reaching to the detector from the to the analyzer. So that is what we are getting a particular top. So now what is happening is that so if you have the singly charged ion species and it will be in a range will be pretty high. So range is pretty high. How you can understand, right? So if you have the two charge, it will be, so for instance, it is 80. Okay, so 80 plus 1 by 1 equals to 81. But if you do here 80 plus 2 by 2, that is equals to 41. So you can understand that the range becomes reduced when you have a doubly charged ion species or triply charged ion species. Then what it happens is that your particular range gets reduced. You can uh, you can have your particular mass analyzers that is up in a lower range. But if you are using of only singly charged species, then your ranges will be pretty high and your range will be pretty high. So that's why if you are doing for the top thing, Maldi top, and you are generating on the singly charged ion species, then what it will happen is that you need to have your top mass analyzer. So this top mass analyzer to be pretty long. Okay, it will be pretty. So it will be pretty long, and you will not be able to get those uh, particular but it will be a huge space issue for you to have and then to manage and all those things. It will be a kind of a logical constraint, and this particular thing will be used with a huge difficulty. So that's why the MALDI has its limitation of the singly charged ion species, and it cannot be used in a wide variety of proteomics application just because of its limitation in creating ions and how much it is making up. Okay, so now there comes the principle of ESI, which we were noticing is the electrospray ionization. So here you can see that your particular ions will be in the gaseous form, and through the particular this cone that is known as your Taylor cone. So this is your spray needle, and through the application of this power, electric power or voltage, okay. So through the use of this electric power and the voltage, so you will have those particular ESI solution, which will lead you to the droplets and then finally go through the mass analyzers into the mass spectrometer. Okay. So now if we talk about the principle of ESI, so there are two models. One is ion evaporation model and one is the charged residue model. Okay, so there are two models for the dig deeper into the principle of ESI. So what is ion evaporation model? So it was proposed by Ribben and Thomson. So it suggests that the ions are ejected directly. So how the ions are ejected? That the ions are ejected directly from the surface of charged droplets, and here as the droplets shrinks, so the droplet will be shrinking, and the ions are ejected directly from the surface. So this solvent, what happens? That solvent evaporates, the surface tension competes with the increasing Coulombic repulsion until ions dissolve from the droplet into your gas phase. So we know that we have talked about that your particular ions are in the gaseous phase for the electrospray ionization. So it we are telling that the solvent will uh, evaporate and the surface tension will compete, and increasing there will be Coulombic repulsion, which will lead you to the ions dissolved from the droplet into your gas phase. So this is ion evaporation model is more applicable to smaller highly charged ions, whereas the field strength at the droplet surface is sufficient to your for your expelling the ions. So this explains the ionization particles for smaller molecules and peptides, which are relatively low molecular weight, and its critical low role of electrical fields droplet surface in enabling ion release without breaking your droplet apart. And then another model is the charged residue model. So it was given by Dole and then by Rolgen and other uh, refined it, that droplets continue to evaporate. They sequentially break into smaller droplets until only a single analyte ion remains. So it will continue to evaporate, and finally only a single analyte ion is being there. The charge is retained on the residue after all solvent evaporates, leaving a gas phase ion with minimal fragmentation. And CRM charged residue model is often considered more relevant for large molecules such as proteins. Complete dissolution leaves behind the charged macromolecules. So gradual stepwise process from Coulombic fission results in droplets in contexts where the analytes are large and require more gradual solvent evaporation before ionization occurs. So you can understand from now from this thing. So whether if your analytes are large complex ions like proteins, so it will be like the charge residue model, or if we are having the proteins peptides, so we will be having the ion evaporation uh, model. Okay, so you can look into it in a much deeper way. So if we talk about the general principle of ESI, of course, these two models will further describe it. But if we did talk about the general principle of ESI, it is the droplet formation. So the sample solution is ejected. So you can understand that it is ejected from the capillary. So under a high voltage electrofield forming charged droplets. So ultimately, if you so that's why in that first slide of the skeleton of mass spectrometry, we have talked about is the creation of ions, then finally separation of ions, and then followed by the detection of ions. So this is what is happening, that it is ejected from the capillary under a high voltage electric field and forming your charged particular charge droplets. Then you have the initial dissolution. So enters the spray chamber, heated dry gas. Okay, so heated your particular dry gas, example nitrogen, evaporates the solvent, leading to smaller droplet and increased surface charge density. Okay, so first of all, if I just do the schematic as per it, so what is happening? So you have the sample solution. So I am denoting S solution. So it is ejected from the capillary, ejected from capillary under high voltage electric field. So sample solution which is ejected from the capillary under your high voltage electric field, which forms your charged droplets. So it is producing your charge. So it is producing your charged droplets. So then what we are going to do is that these charged droplets, when it enters the spray chamber, so you have the spray chamber, there will be, so nitrogen gas evaporates the solvent and your droplet size will continue to decrease and increase in the surface charge intensity, because the surface is getting decreased but the charge is there, so it will be that will get much more charge. So first of all, you can imagine now that you have the solution. So it was ejected from the capillary under your high voltage, forming charge droplets. Now, when you are going to it, so this is the process is also known as a desolvation. So this droplet, when it is entering the spray chamber, so what is happening is that heated dry gas, that is the nitrogen, evaporates the solvent, leading to smaller droplet sizes and increase in your surface charge density. Then what is happening is the Rayleigh limit and Coulombic fission. When the droplet size reduces and charge density reaches the Rayleigh limit. Okay, so this Coulomb repulsion counteracts surface tension to undergo fission into smaller, more highly charged droplets. Okay, so first step is your droplet formation. So that is the sample solution is ejected from your capillary and under high voltage electric field, it is forming your charged droplets, then followed by initial dissolution, that is your particular droplets will enter the spray chamber and heated dry gas, that is your like your nitrogen, that will evaporate your particular solvent, and it will lead to smaller droplet sizes and increase your surface charge density. So then it will keep on continuing to it and it will reach the Rayleigh limit, where this repulsion will counteract and it will undergo much more greater fission. So it will be continued dissolution. The cycle of dissolution and fission continues, concentrating charges onto the remaining droplets. Finally, gas phase ion at the nanometer scales, ions in the highly are eventually released into the gas phase, forming gas phase ions ready for mass spectrometric analysis. Okay, so this is the basic principle, that how that particular electro spray ionization is happening. So if we talk about, so electro spray ionization, so this is how it is sprayed, forming the charged surface. Then nitrogen gases to it, then you will reach the Rayleigh limit, where it will pass further, and it will continue to do it, and then finally ions are highly and released into your gas phase, that forms the gas phase ions ready for mass spectrometric analysis. Okay, so hopefully you don't have any question right here. So no questions, right? Okay. So you understood, right? Principle of ESI. Okay. So now, as we mentioning that the time of flight mass analyzer. So now we will be looking into it in short, the different mass analyzers that are present, that how the MBISs we will be getting those particular MBISs, and how we are going to analyze it, right? The finally the detector through how it is going to affect. So now first of all, it was getting difficult for us to control the ionization procedure. Then we could come up by lowering and controlling the ionization procedure. One is through our MALDI. So one is through our MALDI. Yes, that is the sample is injected into the matrix, and then the laser uh, that is irradiated, and finally ions are forming, and then is the electrospray ionization or ESI, where the charged droplets are uh, released through the capillary and then going through the desolvation, where the ions will be formed, and then they will reach the Rayleigh limit, and further it will go on, fission will happen, and it will form the charged droplet ions. So now this thing is getting ready to be injected into the mass spectrometer, right? So before that, also one step is there, that is from the peptides we need to do a separation, fractionation, but we will be taking into it in a separate aspect in shortly. But what we are doing is that once our particular uh, ions are ready after the continued fission and separation, as we have said, that the highly charged droplets are eventually released into your gas phase. So what we are doing is that we are releasing our particular thing into the gas phase ions, so ready for mass spectrometry analysis. So then this comes the mass analyzers that are that's basically having the detectors which are going to detect that which particular ions are coming. What is the how to calculate that now? So among which one of the analyzers that is being used widely is the TOF, and I have spoken very briefly about the TOF when I explained you about that the multi. Some of the limitations of multi is that if we are coupling multi with TOF, multi TOF MS, and if it is only the singly charged ion species, which is resulting from multi, and if we have a greater number of length of those particular peptides, then it will need a very long particular uh, time of flight to detect the time taken by those to reach to that particular detector, right? So now how this time of flight works is that you have the ionization area that will be taken through acceleration, and after that heavy ions and light ions will drift and it will be detected. So this separates our ions, and what it does is that it measures their imbices based on the time they take to fly from ion source to detector. So this is the way through which our ions are getting uh, through which our ions are getting from the source to the detector. So this is how the ions get separated. So this measures the imbices based on the time they take to pass from the ion source to detector. That is the time they take to fly from the ion source to detector. So this is how it calculates the M by Z. So we will be doing some problems on the calculation of the M by Z's ions about the different time taken, or the distance, or the that how much the time of flight's distance would be, or how much time is taken for an ion to reach on the given certain condition. So we would be looking into it in some time. So this is one of the mass analyzers, that is time of flight at TOF, which is used heavily. Next is the quadrupole. So after having the TOF, now we are moving towards different other mass analyzers that are being incorporated into different high resolution mass spectrometers, and that has increased the coverage and depth of the identification and quantification of the proteins to a great extent. So quadrupole is something which contains, so from the name, it is quite clear, right? So quad coming from four, right? So these four are the rods. So there are basically four rods. So two opposite rods with positive potential and the two rods with your negative potential. So two of you have positive potential and two rods have your negative potential. So what this is doing, this particular positive potential and the negative potential, so this acts as your filter, right? So this acts as your filter which allows all the ions of a certain mass to charge ratio to a certain filter pass to this filter. So what happens is that when so now there are different high resolution, high end mass spectrometers are generally hybrid mass spectrometers. So why? Because these enables these hybrid mass spectrometers. What it does is that if we talk about this particular mass spectrometer, so these help us. This combination of these two mass analyzers help us a better separation of ions. So the more the better separation would be, the more amount of ions we would be getting, and we would be identifying in our the coverage would increase to that extent. So that's why it becomes quite important to have different use a combination of mass spectrometers so that effective separation can happen. So quadrupole is something which is used heavily, like Q. If you go and search, there you will find QTOF, that is quadrupole mass analyzer is coupled with time of flight mass analyzer. So you can have quadrupole to be matched with orbit trap. So here what happens is that it contains our two opposite rods, one with your positive potential and another with your negative potential. And what happens is that it will create certain filter that it will allow certain mass to charge ratio to pass. And in this way, it will lead you to the better separation and effective identification of proteins or peptide ions at the end. Next, as I mentioned, that it is orbit. Another thing is orbit trap instruments. So these mostly come in hybrid configurations. So hybrid configuration, you can say that there are two mass analyzers. So it is coupled with, for example, quadrupole. And what is happening is that these ions are separated in an oscillating electric field. So you have an oscillating electric field. So ions will get entered. So for example, if I say that you have quadrupole, so you have particular four rods, right? Four rods that is filter. So this will separate the ions, that is separate some ions to pass. Now if you it is coupled with orbit trap, right? So now if it is coupled with orbit trap, so here it will ions will separate uh, again to an oscillating electric field and will reach to C trap. So C trap is something which will store the ions for such some short period of time, and then what it will do is that it will allow to go to the detector for detecting it. So this is classical example of using hybrid mass spectrometer where quadrupole is coupled with, for instance, orbit trap, and then finally the role of slit trap in storing the ions and to what extent, and then finally detecting. So finally, it will give us the detection in the mass spectrometer. So basically, what we are doing is that we are trying to separate the ions in as much possible way so that our final separation goal is quite achieved in a larger extent. So another mass spectrometer that is there, that is the ion trap. So that is also similar to quadrupole, which taps the ions and selectively based on the M by Z charge. So this is again another which is the ion trap. So now for tribrid mass spectrometers, you can have configurations like for tribrid mass spectrometers, you can have quadrupole, you have orbit trap, and you have can have ion trap in the same instrument, and you can use any two at a time for separation and detection. Okay, so this is also the one that helps you to collect uh, trap the ions and then selectively pass your ions so that you can do it for your the basis of your MIC charges. Okay, yeah. So this is what we talked about, right? So any question or any doubt regarding those particular thing? Is it clear, right? Okay. So there is we'll do some of the problems with the time of flight mass spectrometer. At least we will see some basic problems that how you can calculate the distance required for the uh, particular if your ion you want to detect in a particular time as it for effective separation, as well as if your ions can be reaching at what particular time, and all these calculations we will be doing in through our we will see some of those problems, kind of problems how to solve. Now the term comes. So I have talked about in the beginning only that we had seen that there was written that MS spectra, right? MS. So what is that? So we are calling it as a tandem MS. So what we talked about is that we have peptides. So we also gave an expression example. So AAV, whatever be a peptide, you can take arbitrarily any peptide. So if you are taking arbitrarily any peptide, and if you are saying that its molecular weight, for example, is 304, uh, so 304 Dalton, for example, I'm just taking an arbitrary number. Similarly, another peptide, if you get like T, BL, DA, something like that, which is also containing the similar same amino acid, uh, same uh, uh, same molecular weight, right? So now this has also same molecular weight. This peptide too has also same molecular weight, but we cannot. But this belongs to, for example, protein A. This particular sequence has a different function, different structure. Protein X has a different structure. Protein X has a different function. Similarly, protein Y. So protein Y has a different structure, different function. So protein Y has also a different structure, different function. The sequences are different. The way they are written. So if that is of the same amino acid also, but the sequence order is quite different. So they do belongs to the different proteins and work and helps in the structural and functional association of these two different proteins, which is quite different from what is generally seen, right? So now how to do that and how to analyze that and how to identify those particular peptides, that is actually this is contains this particular sequence, and it is not the other sequence, or how to deduce the sequence of the peptide. So this is where the concept, sorry, of tandem MSMS comes. So what does it say? Is that you have the sample. So you can have ESI MALDI, will have the ionization, you will have them by the separation, and you will get, so this is the sorry, if you whatever you are getting the molecular weight, calculating the molecular weight. So if you are calculating the molecular weight of the particular peptide, so that you are talking about precursor, right? That is the total peptide one, for example. This is the molecular weight we are getting. But what is the sequence? So if there is, what is the sequence, that remains a question mark. So for deducing the sequence of the peptide, the concept of tandem MSMS is comes up, emerges, and it helps us in deducing the actual sequence of the peptide. So how it does? So it will take down the, so it will further this particular sequence. So for example, this sequence, these will be again fragmented. Okay. So these particular sequence will again fragmented using collision surface. So this is the case. This particular peptide, once we are getting the molecular weight, we are further fragmenting it to get the MS second level of uh, ions, second level of fragment ions, which will be then further taken for the M-by-the separation and the detection. So this is known as the precursor ions. This fragmentation of those particular peptide is known as your product ion. Okay, so this is how after the fragment we will be getting, because we will be using a specific energy. So here what we will be using a specified specific energy, right? So this will have an unique fragmentation pattern. So it will have an unique fragmentation pattern, and through what we can reduce it. So in the first particular slide, we had shown that there is Y ion and V ion. So we can minus each ions, which will give the monoisotopic mass or a atomic mass unit of those particular peptide, particular sequence of amino acids. Okay, so this led to the term coming of tandem MSMS, or MSMS fragmentation, or MSN fragmentation, that you could see, that is the MS2 level of fragmentation. Then what we are uh, talking about is the precursor ion and the product ion. So this what we are telling, that MS1 can help in identifying our molecular weight of the biomolecule. If you have the MS1, can help in your identifying molecular weight of a biomolecule, and MS2 is required for deducing the particular peptide sequence. Yes. Now in this tandem MSMS, what we were talking about in beginning only, I have spoken about is that bottom up approach and the top down approach. So what is bottom up approach is that we are taking the whole protein or proteoforms and we are breaking down into pieces. Breaking down means we are doing some enzymatic digestion. So we are basically breaking down into pieces. So we are breaking down into pieces and then we are identifying it, particular peptides, and then we are finally pressing it back with the protein. So aligning with the protein from which protein it belongs to. So that's why it is known as your bottom up approach. Whereas in the top down approach, you will have an protein set, entire protein set. You will do the electrospray ionization. You will have the intact precursor ion fragmentation, and those fragment ions we will talk about the protein. So that's why we are talking about the bottom up approach, as I had mentioned in the beginning itself. So we are talking about the bottom up approach. Yeah. So any question till now? No, right? So it's clear, right, that tandem why 2 ms fragmentation is required. And then we are talking about the bottom up approach and the top down approach. Okay. So this is the tryptic digestion, ionization, and then finally we are going for the electropeptide ions fragmentation and then identifying it and then finally getting this particular peptide. So now in this acquisition method, so I'm not sure whether in this week have you gone this through two terms, data dependent acquisition and data independent acquisition. Okay, okay. So I'm not pretty sure that it has gone, it has been there or not, but yes, definitely it will be coming in some if it is there in this particular week or in the next week. So what we are talking about, just understand. So here it is name comes from data dependent acquisition. And what one we are talking about is data independent acquisition. So basically what we are saying that we are taking the M by Z's, that is after ESI we are doing the in the tandem MS, if you go see the ionization and you are doing the MS1 separation. So this is our MS1 separation ion. Now if you just take that top N abundant ions or top N or that N can be 10, N can be 20, N can be 30, etc. So that those you will be performing the fragmentation of these ones only. Okay. So you are going to do the fragmentation of the selected precursors. So you are performing the fragmentation of those particularly higher uh, those abundant intensities precursors only you are going for the fragmentation and you are seeing at the MS2 level. And in the data, so this is becoming dependent in a sense that you are taking top in abundant precursors that is happening here. So this is what it will be. It is taking those particular ions and then particular intense peaks, and that you are fragmenting it. But in data independent acquisition, what is happening is that you are, for a given embodies window, for example 50 to 1500 embodies. Okay, so what you are doing is that whatever ion comes, be it less abundant, be it high abundant, anything, all you will be doing the fragmentation of all the precursors in each window. Okay, so this is the two acquisition methods. One is for the data dependent acquisition in the tandem MSMS, that which precursor peaks are you going to fragment it, depends on your intensity levels or the abundance levels. And for the data independent acquisition, what you are going to do is that you are taking the entire window spectrum of the spectra for a given particular window, and you are fragmenting each and every one. You are not seeing any intensity, which one is the abundant one, which one is the lowest abundant one. You are not going to choose any of those particular things, and you are completely going on for the fragmentation. So this, what it does is that if you are doing this particular kind of fragmentation, so you are not depending on your particular knowledge of your intensity, and you are just uh, going through by fragmenting the entire window. So that's what gives the name data independent acquisition and the data dependent acquisition. Okay. So now if we talk about, there are certain parameters which we need to keep in mind and understand that how we are going to do is, so that one is the resolution, isotopic peaks, and the charge state. So what is the resolution? So here you can see that if you can, uh, so the so resolution by uh, literal meaning or by definition is that ability to differentiate closely related things, right? So this is what resolution means, right? So anything which is very very uh, yeah, so anything which is closely related, if you want, if you can separate it or segregate it, so that is what termed as a resolution. So now if I talk about that, here you can see that, for example, here it is written. So here it so just a minute, I explaining it. So m by z difference between isotopic peaks is here 0.25. So 1 AMU, so 1.254. So if there is 431.73 into 4 minus 4. So you can see if it is resolution is 60,000, example in a orbit trap, you can see 1 peak, 2 peak, 3 peak, 4 peak. So all are distinct peaks having very close approximation of the embodies. That is 432.73, 432.48, 432.23, 431.98, 431.73. So you can see that each and every closely related uh, embodies are can be very very peaks are deconvoluted, and you can get sharp peaks. But if your resolution is low, you can see that you cannot get a single sharp peaks for each embodies, and it is fully for the 432 embodies you are getting this kind of peak, right? So this kind of peaks you are getting for 432 embodies, whereas if you have in the fourth in this range only, if your resolution is high, you can have 4 different peaks. So this will definitely lead you to the better resolution and better identification of peptides. But here it will be not. So what is the by definition? So it is that the mass by delta mass. So dimensional ratio of m by z value of a peak divided by its width at half maximum intensity. So what is the at full width at the half intensity maximum? What is the uh, range, that is delta mass and divided by mass. So this will lead us to understand that if your peak is coming these and these, so whether these two peaks will be able to resolve or not depends upon your mass resolution. Now the next thing, so is it fine, right? So you have understood the meaning of resolution. Okay. So now if we move ahead, so next term comes the mass accuracy. So why mass accuracy? So whatever we are identifying, so exact mass and the experimental mass, what is the difference between the observed mass and the theoretical mass, that gives us the actual accuracy, because it the whatever is the theoretical mass of a particular peptide or a particular molecule or the those things, we tend not to achieve those exact mass by virtue of the experiment. Rather, the average there appears to be certain experimental errors because of the sensitivity of the detection of the instrument and others. So it, of course, it leads to have certain changes in the particular mass accuracy, right? So this observed mass minus the theoretical mass upon the theoretical mass, when you multiply by 10 to the power 6, you will be getting the mass accuracy in ppm. So if you want to get the mass accuracy in ppm, how we will be doing is that you will be getting the exact mass and the average experimental mass. So when you give minus it, the observed mass minus the theoretical mass, and then divided by the theoretical mass into 10 to the power 6, you will be getting into the ppm. And similarly, you can calculate yourself to get it ppb and other units. So now we had talked about the tandem MSMS, and we had talked about the different techniques of acquisition techniques, like the data dependent acquisition and those data independent acquisition. Now, if we talk about that, how this fragmentation from the MS1 to MS2 occurs and what are those types. So we have talked in great detail. So if I again come to this particular picture, so we have talked in great detail about here ionization of AI-SI multi. And now what we are talking about, that after this, either we will be choosing top, like you can think that you can will be choosing top in ions, in abundant ions, or all ions in the window, right? Or all ions in a particular window you will be choosing, and you will be leading to the fragmentation, right? So now these fragment becomes crucial. So what are the how that fragmentation happens? So that fragmentation cannot happen, or that ionization cannot happen like the way has been done before. So here it is being the collision induced dissociation if it happens. So if the collision induced dissociation, if it is happening, so then it is the so what is happens is that from the name it is quite clear that what we are trying to say is that collision It induces the dissociation through the collision. So collision with inert gases or noble gases. So what happens is that the precursor ion, either you are doing it through uh, EDA, that is through the top n abundant ions, or through DIA, that all ions. So what you are doing is that there is a collision gas, and those collision gas are going to collide with your precursor ion. So this is the term given, that is the collision induced dissociation, which leads you to have the product ions. So from the precursor ions, you are having the product ions through collision induced dissociation. Finally, you are getting the MS2 ions. So from the MS1 to MS2 with the help of collision gas, which are generally your inert gases like helium, or those things are utilized, and this finally leads you to the getting the particular MS2 level fragment ions. Okay, from the MS1 to uh, MS2. Okay, so this is one. Another method is that electron transport dissociation, that is the peptide cation which you will have. So that you can take it, and you can have the excited charge reduced ion and then proton transfer product, and this is through the electron transfer dissociation. But generally that is being followed, that is being different HRMS, that is high resolution mass spectrometers, are employing is the collision induced dissociation, where you will be using noble gases and inert gases to fragment your particular ions, precursor ions, into the product ions, MS1 to MS2, and finally will be deducing the length of those particular peptides. Okay. So now we will be understanding the chromatogram, but before that, let us first understand the gradient. So what is the gradient? So how? So you had a question, right, about RPLC. That what is the meaning of RPLC? So what we are talking about is the reverse phased. So we are talking about reverse phase liquid chromatography. So what does it mean? So it means that the increase in hydrophobicity to elute out the hydrophobic peptide. So what is the principle? If I talk about RPLC in general, reverse phase liquid chromatography. So you have, for example, you have the chromatographic column. So you have your particular, this particular column is bound with your hydrophobic uh, material. So for example, C18 is one hydrophobic material, that is carbon with 18 isotope. So what it is, if what you will be doing, so you have your complex mixture of peptide. Okay, just listen it carefully. So you have your complex mixture of peptide. Now you want to separate your peptide mixture so that it becomes less complex, right? So your peptide mixture, you are wanting it to pass through it and get it fractionates out, and you want to get it to become quite less uh, what I can say is that you can want to have it as a you want to separate it. So what happens is that if you, so peptides are hydrophobic by nature, so they are your hydrophobic by nature. So once your peptides are hydrophobic by nature, then what is going to happen is that when you pass this peptide solution to this hydrophobic mixture, so this peptide solution you are passing to it, so what it will happen is that your in this particular hydrophobic material, peptides will get stuck. So peptides will get stuck in these artificial materials. So these are generally happens through our analytical columns. So these are all packed with the hydrophobic material like C18. And once these are all packed, so what happens is that once this particular uh, things gets packed in the hydrophobic, so these peptides, what it gets is that it gets stuck. Now, what happens is that when we will increase the hydrophobicity as we move along for making these peptides come out through the solution to elute out on the basis of their hydrophobic nature, we will increase the hydrophobicity, and generally the solvent that is being used is that the percentage gradient of acetonitrile is increased. So that is what we are talking about gradient, that at what percentage, at what rate we are changing the hydrophobicity percentage of acetonitrile or ACN so that it keeps on increasing the hydrophobicity and our particular peptides are being eluted out. Our particular peptides are being eluted out, right? So how it is done is that the flow rate, so generally for the effective separation of the peptides in proteomic studies, what we do is that the flow rate is kept in nanoliters per minute. So it's a very low flow rate happens so that very less amount of peptides gets eluted out. And then finally, after we are having an effective uh, particular ionization procedure, then we get to that particular protein. So once we are done with the water ACN, so what happens is that in the reverse phase liquid chromatography, we will be increasing hydrophobicity to elute the hydrophobic peptide. So what happens is that the peptides gets eluted. That is, the less hydrophobic peptides will be eluted, followed by more hydrophobic peptides because we keep on increasing the gradient. Okay, so this is how this is performing. So that's lead us to this particular chromatogram. So as and on when, so this is chromatogram is coming from the ions. Okay, whatever you can total ions chromatogram, that is the peptide as on it keeps on eluting it out. So what happens is that when the peptides gets eluted particularly out, so what happens is that so here it is 120 minutes. So you can see the time here. So 0 to 120 is the end time. So once you have the end time, so your gradient, as I have mentioned, that you are going to be increasing the pattern, increasing the hydrophobic, increasing the percentage of hydrophobic, and your peptides will get keep on from your column, and it will be then perform the ionization using the principle of ESI as we have discussed by forming the solvent and droplet ions. And as it moves along, then what it is going to happen is that the chromatographic separation of the peptides will keep on happening for 120 minutes. So a good chromatogram should be a Gaussian pattern and the intensity is in the range of E9. Uh, and what will happen that illusion static point and end point is crucial. So there should be no humps or distorted peak. Otherwise, it will show that some contaminants from the previous run or some issue with your analytical column uh, has eluted out suddenly with humps and those kind of distorted peak are kept in mind. So here, for instance, you can see this particular chromatogram. Okay. Are you getting it right? Uh, is everything is clear, right, about the gradient and the chromatogram? Yeah. So now You can see this particular chromatogram is there. So here you can understand from this chromatogram is eluted to over the period of time with the maximum eluting in this range of those hydrophobicity. But here you can see the dilution started, but it got failed in between. It can be issue with the sample, issue with the column. So here again in this chromatogram, you can see that the chromatogram is not so Gaussian in nature, not so dense, so protein coverage would be so great. We can understand, right? Talked about this gradient, and that's why we are getting this chromatogram of ions as on when the hydrophobic peptides are getting eluted out, we are finally getting the what is the measure of the ions, and then finally we are detecting what is detecting it in the detector. Now there are certain concepts related to it. One is your dual volume. So what is dual volume here? That is the system volume. So from the point are mixed. So here, as we have said, that we are using two solvents. One is your water, and another is acetonitrile. So here, whatever is there, that the mobile phase solvents are mixed until they reach to the head of the column. Okay, so your solvents gets mixed and then it reaches to your column. So what is that volume that is required? So this is your mixer, connecting tubing, auto sampler loop as your uh, different primary components. Okay. Now then in another thing, what is there is the dwell time. So what is dwell time now? So it is obtained by dividing your dwell volume by the flow rate. So why it is very much important, because it will lead you to understand that how the mass spectrometer can go and how much peak width will be there. That will take how many data points. So maximum data points means the peak is very robust and sharp, and it will be uh, fitting into the, because as many as data points it will be collecting it. To form the peak is easy. For instance, here you can see that it is very distorted peak. Uh, uh, Data points, that is not very effective and very efficient identification you can talk about. So a system with a dual volume of 3 ml run at a flow rate of 1.5 ml per minute would have a dual time of 2 minutes. Okay. And delay created by the dual volume can make a difference in the appearance of the chromatogram for different gradient systems. That's why it is very important. So this delay created by your dual volume can make your difference in the appearance of the chromatogram. So no effect on isocratic flow where the components of the solvent mixture remains the same. Okay. So this is the two concepts of your dual volume and dual time, which leads you to the effective chromatogram and efficient peak identification, which is quite crucial when you are doing. Okay. So now if we move on, as I mentioned that we will be discussing certain aspects of calculation specifically for the time of flight mass spectrometer. So we talked about the time taken to travel distance. So this is basic physics is involved in it. That how to calculate the time, how to calculate the length of the tubes, and those things. Quite briefly, the principle that is followed, the formula that is followed, you can go, you all can go back, and what you all can do is that go and practice this kind of sums. It will help to understand. So what we are saying, that we are taking like the time by your distance by velocity, simple. So we have the equation for velocity is that root over 2 k by m, because of half mv square equals to kinetic energy. Now, if we substitute this into t into distance by velocity, so that will be distance by root 2 k by m. and if you rearrange it, it will be d root over m by 2 Ku. So, for example, if we talk about mole of Mg2 plus ions has a mass of 24 gram per mole. To find the mass of an ion, we need to divide by Avogadro number and then divide this number to convert to kg. So the time to reach for 1 mole uh, will be is that uh, so you have the one distance is given as 1.2 meters. So mass but, given, you will be calculating it and then you will be giving into the time. Okay. So how this mass is generally calculated is relative isotopic mass into 10 to the power minus 3 by 6 by Avogadro number, and kinetic energy, as we have said, that is half mv square 2 k by m, and velocity is distance traveled per time. So 56 SR plus, that is the strontium ions, travels to the top of a mass spectrometer with an energy of 1.92 with an energy of 1.921 into 10 to the power minus 2. That is, you can calculate the kinetic energy is given. So it takes 1.09 into 10 to the power minus 5 second to reach the detector. So the time is also given. So then, so here there is kinetic energy to calculate the length of the flight tube in meters. So you will calculate the velocity, and once the velocity is calculated, you will be substituting in this formula, that is distance by time, and the time is given. So you will be length of the flight tube in meters can be easily calculated. Just you need to be careful in looking at the particular units of those kinetic energy and those particular distances. And you're done with it, you can easily go and search for understand that what is the final uh, value which you will be taking forward for calculation for the length of the distance and calculating those things. So this is quite straightforward in understanding, and of course you can go back and you all can go back and also perform calculations related to it. But these are some of those basic things which I thought of making you aware that this works on the basic principles of physics through which we can easily calculate our results or data. Yeah, so this was all for this session. Hopefully you have understood it and enough. Otherwise, we all can again meet on the next week with the further concepts on the proteomics uh, with the data analysis and other things. And of course, you can see that where we can incorporate different hands-on and other extra things. Okay. So any questions? Okay. So thank you all for joining this session.